Hello, and welcome to ResearchPod. Thank you for listening and joining us today. In this episode, we will learn about how an international collaboration of researchers, including Joaquin Hortal and Juliana Strop at the National Museum of Natural Science in Spain, Richard Ladle at the University of Porto in Portugal, and Gesiane Tessarolo at the State University of Goiás in Brazil, are constructing the first maps of biogeographical ignorance, or MOBIs, that account for data-driven uncertainty in biodiversity analysis. Presented alongside species distribution models, MOBIs can help conservation resources to be appropriately distributed. In a technologically advanced society where data can be collected remotely, almost continuously, and at a low cost, biodiversity research has become increasingly data-driven. Biodiversity data, that is, records documenting species occurrence, can be readily collected from field surveys to assess the relationships between biodiversity and the environment. Scientists, conservationists, and policymakers use these data to understand where species are, why they are there, how their distributions might change under future climate or land use scenarios, and how these changes may affect ecosystem services. They can use the data to reveal the various benefits ecosystems provide to mankind, including food and water, and for improving human resilience to extreme events like droughts or floods. With this knowledge, impacts of global change on biodiversity can be forecasted, and species and habitats better protected. However, conservation science is a crisis discipline, and data collection is not always able to keep up with rapid decision-making requirements. Old, limited, and incomplete data are frequently used as the best information available in times of need. The challenge is even greater when the data is required over particularly wide-ranging or long-running scales, due to the limited resources available to biodiversity research. In the absence of consistently reliable and widespread observational data, scientists rely on methods to estimate distributions from smaller amounts of data. The most widely used are the aptly named species distribution models, or SDMs. SDMs are mathematical models that relate the observed distribution of a species to a set of environmental predictor variables, such as climate, habitat type, and distance to water. Despite their widespread use in species and habitat conservation, these models contain an unavoidable degree of uncertainty associated with the predictions. The accuracy of any analysis or prediction depends on the quality of the original data. In a paper published in 2013, two researchers identified three major factors that compromise the quality of species distribution knowledge at any given spatial scale. They are Joaquin Hortal at the National Museum of Natural Science of the Spanish Scientific Council, CSIC, and Richard Ladle at the Research Centre in Biodiversity and Genetic Resources Associate Laboratory, the CBio in Bio, which is at the University of Porto in Portugal. The three major factors they identified are survey completeness, the decay of information with time, and the decay of information with space. These decays account for the progressive loss of value of data to predict biodiversity trends at an increasing distance from the observations. Survey completeness can be impacted by the difficulty of detecting species due to their characteristics, such as cryptic coloration, elusive behaviour, or high mobility, and the characteristics of their habitat. As a result, even surveys employing a range of sampling methods can be incomplete. Hortal, this time working together with Juliana Strop from the Spanish National Museum of Natural Sciences, Tadeu Sobral, and collaborators from several Brazilian universities, found that sampling sites for butterflies in the Brazilian Atlantic forest were biased towards large and connected forest fragments. In an SDM constructed with incomplete survey data like this, the inference of the functional relationship between butterflies and deforestation would be limited to these well-sampled areas. Increasing sampling effort in small and disconnected forest fragments would lead to more accurate evaluations of landscape scale effects in the future that can be used by conservation decision-makers. A similar study on Iberian mosses found that there were also spatial gaps and biases in the dataset, and dates were lacking from the metadata of many records, further limiting analysis in temporal biodiversity shifts associated with global change stresses. The decay of biological information with time and space comes about because species are continuously responding to ongoing environmental changes. Changes over time include climate change, land use change, habitat degradation, and biological invasions. For example, by 2017 in the Brazilian Amazon, 30% of all locations where tree specimens had historically been recorded had been lost to deforestation. A further 300,000 square kilometres of rainforest had been deforested by 2017, without having a single tree specimen recorded. Lack of historical data about which species occurred in deforested areas presents a particularly grave problem because this type of information is irretrievable. Changes in space include climate, habitat type, soil chemistry, level of disturbance, and community composition. As such, the certainty of SDM predictions decreases as the spatial and temporal distance between data collection location and a modelled area increases. To maximise policy and conservation impact, maps of data-driven uncertainty or inaccuracy within SDMs should be displayed alongside the outputs. Research by Hortal, Leidl, Geisiani Tesarolo at State University of Goiás, Brazil, and colleagues produced the first maps of biogeographical ignorance, MOBIs for short, that do just this. They account for survey completeness and quality of data in each area, as well as the decay in information with time and spatial distance. Highlighting these uncertainties helps identify future conservation priorities. They can determine areas with more reliable predictions and support decision-makers using SDMs, for example, in the allocation of future conservation priorities. Regularly updating the combined distribution and ignorance maps would provide users with the information required to deal with biogeographical uncertainty through the design and implementation of new surveys, or by incorporating spatially explicit estimates of uncertainty into future SDMs. A combined approach would also allow conservation decision-makers to prioritise the existing conservation needs of species and allocate resources effectively, for example by focusing resources to survey and conserve the biodiversity of areas that are vulnerable to deforestation or climate change but remain poorly sampled. To produce MOBIs, the research group combined analytical and visualization tools to represent four main sources of biological ignorance in a Scarabaeidae dung beetle dataset from the Iberian Peninsula. They assessed data completeness, taxonomic quality of data, temporal decay in information, and spatial and environmental distance to the adequately surveyed sites. A biogeographical ignorance value was calculated for each record, generating a MOBI for each species. This ignorance value represents the degree of reliability of the biogeographical information for a species in a location. So, the higher the value, the less reliable the information in the area is. A biogeographical ignorance value for the focal species is attributed to each cell of the studied area, depending on whether the cell contains original data on the focal species, a species from the studied taxonomic group, or neither of these. If the cell contains a record for the focal species, the biogeographical ignorance value is taken from the most reliable record. If the cell contains a complete record of the species of the same taxonomic group, species that can be captured with similar sampling protocols, the absence of the focal species is likely true. So the biogeographical ignorance value is taken from the mean minimum of each species in the cell. For cells without data for the studied species group, Environmental and spatial proximity to cells with data is the only way to calculate ignorance. The researchers combined SDM maps with MOBIs to visually represent the reliability of distribution knowledge. Both species distribution models and maps of biogeographical ignorance produce maps displaying continuous values. For easy interpretation, the study described 4 extreme combinations of suitability and ignorance, categorized as high or low. The first is areas with high prediction suitability for the focal species to occur and high ignorance. Secondly, is areas of high suitability and low ignorance. Thirdly, is areas with low predicted suitability and low ignorance. And finally, the fourth combination is areas of low suitability and high ignorance. In terms of conservation, priority based on these outputs, areas of high ignorance would benefit from further surveys, and areas of low ignorance could be targeted with conservation management to increase suitability or preserve habitat if they are predicted to be home to the focal species. Ignorance maps that are combined with SDM predictions generate spatially explicit maps of uncertainty and reveal insights into the reliability of such commonly used maps. Neglecting this uncertainty can affect SDM interpretation and thus potentially incorrect theoretical or practical applications, including ill-advised conservation actions. By routinely using maps of ignorance or similar techniques, conservation action can become more effective. In a resource-deficient discipline such as conservation science, it pays to understand the data and its inaccuracies before making decisions that could waste valuable resources or lose invaluable habitats and species. The National Museum of Natural Science celebrates its 250th anniversary this year. As one of the oldest natural history museums in the world, and with a staff of more than 80 researchers, it is dedicated to explaining scientific discoveries to the public. That's all for this episode. Thanks for listening, and stay subscribed to ResearchPod for more of the latest science. See you again soon.